Our next presenter is Aaron Bowman. And Aaron, here's the official clicker. The floor is yours.
All right, thank you very much. It's a pleasure to be here to chat with you for a few minutes about the BIC 2.0 proposal that the group assembled with me was looking at. Our focus is on Parkinson's disease, Parkinsonisms, idiopathic Parkinson's. And just to step back from that for just a moment, many chronic human diseases are the culmination of environmental factors, our own genetics, epigenetics as well as part of that, and a combination of that with time, with aging. And Parkinsonism, Parkinson's disease is no exception to this. The incidence and prevalence of Parkinson's disease in this country is not uniform. I think you can see a pattern up on the screen. There's a common theme here today of bad things in the future. So this is sort of continuing that theme, unfortunately. You'll see where we live here in Indiana is in an area of increased incidence and prevalence of Parkinson's disease. Trying to understand the etiology, underlying causes to idiopathic Parkinson's disease has been a very large challenge, in part due to this combination of a complex network of environmental factors, genetic factors, and time. But pesticides in our environment have come out many times across a number of epidemiological studies to look at risk factors, environmental risk factors for Parkinson's disease. And what you see here on the screen is a map of the annual usage rates of various pesticides. These pesticides obviously play a very important role in the generation of high quantities of food, which obviously is important as well for our own survival. But you can see there's also a non-uniform distribution of many of these pesticides. These pesticides on the screen, including atrazine, glyphosate, mancozeb, and paraquat, have been particularly linked in a variety of epidemiological studies to risk for Parkinson's disease and Parkinsonism. I want to point out as well that the pattern that you see on the screen for these particular pesticides is not entirely random in relationship to the pattern of incidence that you also see. You might sort of see, looking at this sort of pattern and the pattern that you see here. And what this shows us, living here in Indiana, that we are in an area of high risk for Parkinsonism. This rate of Parkinsonism is escalating throughout the country, actually throughout the world, as we get a aging population. And so there's this pesticide usage. So Purdue is basically like a ground zero for high risk for Parkinsonism. We are right near the rural populations where many of these pesticides that are risk factors for Parkinsonism lie. The population around our area is in the population at the higher risk. And so we are placed here in the center of this with, I think, opportunities to take advantage of the information we can glean from this to help us better understand the underlying risks of Parkinsonism. But there are a number of research challenges in understanding this interplay between genetics and environment in human disease. I list a few of these here. One, with various model organisms, there are species-specific responses to environmental factors. So you can't always use, say, a rat model to perfectly mimic the effect of an environmental factor in the context of, say, Parkinsonism. You can sometimes, but maybe not other times. Individual genetic differences between all of us, you, the person sitting next to you, we all have different genetic risk factors for disease, and understanding how our own individual genetic risk factors culminates in our risk is a challenge in understanding gene-environment interactions. The majority of genetic modifiers of disease are unknown. This presents another risk. How do you model the effect of a genetic factor if you don't even know what it is? And lastly, a diverse mixture of both risk and protective factors culminate over time, over human development, from very young ages through aging, to bring about disease risk. How do you understand this? This is a very complex issue. So there has the discovery, which received a Nobel Prize for Shinya Yamanaka a couple of years ago, of induced pluripotent stem cell technology, offers one potential method to bridge the study of human risk for disease and basic and translational studies within the labs. And our group consortium got together to sort of bridge this sort of translational and human subjects-based research with bench level and translational level research, utilizing connections of methodology that can link these two. So this technology allows you to take, say, a skin biopsy from an individual, turn that skin cell into a pluripotent stem cell, which is able to be differentiated into any cell of our body. Why that is particularly special is because you no longer have to know ahead of time what someone's genetic risk is, as long as you know that individual might have been at greater risk than another individual, because that entire genome of that individual is represented in that individual's stem cells. In addition, we can turn these stem cells into, as I said, any cell of the body. Up on the screen is some staining for tyrosine hydroxylase and a general neuronal marker, a marker of the cells of the brain, the dopaminergic neurons of the substantia nigra pars compacta, that degenerate and become dysfunctional over the span of Parkinson's disease. So we can, from an individual in Indiana, take a biopsy and make dopamine neurons, the very neurons that are vulnerable to this disease, here at Purdue to try to study the underlying risk factors of that individual or of the populations that we're studying. So the proposal brings together 5 components. We assembled a very large team. I'll list the team at the very last slide. This is the second to last slide. We're almost through this. I want to step through this very carefully. So it includes a human health assessment cohort. This is to go out into the clinics, go out into the field, into the rural area around us, and to identify Parkinson's patients and families that have increased risk or decreased risk. You can imagine individuals that are exposed to a number of things. Some individuals might come down with PD and others might not. What is the underlying differences that can we make and model those differences in the lab? We need to do exposure and toxicological assessment. We have the resources and expertise here at Purdue amongst a number of faculty to be able to assess and quantify exposures in our environment and exposures in individuals. So this is another part of the team. And then combining these, sort of bridging those to molecular and neuropathological and neurotoxicological assessment, wet lab, bench research, as well as genomics and genetic analysis. To sort of bring these together, we have individuals that will identify, we can make model systems with those, study the actual factors that they were exposed to determine what they were more or less sensitive to in the context of their Parkinsonism. And you can imagine there's any number of hypotheses and questions you could ask depending on the individuals and the population that you're studying. Putting all of this together is communications and engagement with the Parkinson's community. I can tell you, I was just in Knoxville giving a talk to a group of 700 patients and their caregivers. They come out in droves to get information and to understand this long-term chronic disease. I think we can get the same engagement in this population here in Indiana, a place where we know there's increased incidence already. And with that, I will end, take any questions, a list of all of the various colleagues and collaborators that have come together in this proposal, and the numerous colleges and institutes as well that were supportive. Thank you.
We have a question. There's a question right there.
Yes.
Relative news in severe stages of Parkinson's. It doesn't look like you're at this stage in the development of the project, but do you know of any reversals if you take them out of the context of no more pesticides? Do you know anything?
Yeah, so there's been some study on this. Any number of sort of the environmental factors that are thought to contribute to disease risk, any one of these is not like increasing your risk by 100-fold. You're talking about odds ratios of 3, 4, 2 kind of ratios. But nonetheless, for most of these where they've been studies, even if you remove the exposure, the disease processes continue. They set up a neuroinflammatory-like process that's thought that contributes to this. So reducing those doesn't always help. Right now, the best thing you can do if you or a relative has Parkinson's disease, is increase your exercise. That is a resounding yes to being protective at any stage of the disease.
We have a question in the back.
Yep.
It is a clarification. In the second slide, I think it was when you showed the United States map.
Yes.
And then you have a lot of dots in red.
Yes.
I understood that those were the incidence of Parkinson's disease where it's higher.
That's correct. That is correct interpretation.
What is interesting to me that a lot of the reports are more in the
It's on now.
bigger land in the west than in the right-hand side
Okay.
in the east. You see more incidence of Parkinson's disease. So I would expect that if there was a connection then I may see very high numbers.
Make sure you stay.
You know what I mean?
I know what you mean. So there are some areas in the West that incidence is lower. Actually, in those same areas, that map I didn't show, if you look at prevalence, there's some discordance in the incidence and prevalence maps. Some of the large valleys in the California area that use a lot of pesticides also have a large prevalence of Parkinsonism. There's some discordance between incidence and prevalence and that. I don't entirely know if it's clear why there's that discordance. And certainly, as well as the maps that I showed you of a handful of potential risk factors is nowhere near a complete list, right? There's thousands, tens of thousands, maybe hundreds of thousands of potential environmental factors that are contributing to risk. Those were just a subset. And so what we know is that there are environmental factors, presumably, that are contributing to the increased risk in our area. And can we go out into our area, assess what those exposures are, and determine if individuals that are susceptible to those factors living in our environment are the ones that are getting Parkinson's disease or not. So I think sort of get at trying to answer this question in a very complex environment that we live in. I hope that answered your question.
Aaron, thank you so much.
You're welcome.